Oh, got a couple more people joining us. Where'd they go? Okay. So today should be our last day of material before we get started reviewing for our exam, which will be at the beginning of next week. So today we're going to finish up thinking about regulation of ventilation, and then when we meet on Friday, we'll try and tie all this stuff back together, remember what we've been doing for the last month or so. Okay. So when we left off last time, we had started talking about the regulation of ventilation. So now that we know some things about how ventilation works, you know, physically what laws of physics govern breathing in versus breathing out, and how that oxygen and carbon dioxide is moved through our bloodstream, we also want to know something about, like, why do we sometimes breathe faster, sometimes breathe slower? We know we can consciously control that, but it's not always under conscious control, right? Sometimes we notice, we can physically notice that we have anxiety, for example, because we start hyperventilating. So we talk about regulation of ventilation, we're going to be thinking about some of these sort of automated processes that can change our respiration rate. Okay. Still got a couple more people joining us. Okay. That's all right. This is just the intro. Okay. So when we ended on, I have no idea what day of the week it is. When we ended on Monday, I believe, we were talking about control of ventilation, basically by neurons. So we saw that we have some neurons, some nerves that control muscles. They're coming from breathing centers in the brainstem, and we're also going to be thinking about peripheral input to our respiratory centers. So we're going to be thinking about some types of receptors today. Okay. So we saw that we're thinking about controlling skeletal muscles to expand the thorax, right, which we know drops pressure down, controlled by motor neurons. And in particular, we saw that the phrenic nerve is really important, which we've seen in lab. Phrenic nerve goes to the diaphragm, controls breathing. The external intercostal nerves going to the external intercostal muscles, expanding our chest, right? So we're also increasing that volume of the space, therefore dropping pressure down below atmospheric pressure, allowing air to move in. And during forceful expiration, we also recruit the internal intercostal nerves to forcefully squeeze the chest smaller, although we know that expiration is usually a passive process. So we looked at the firing of some of these nerves in quiet breathing versus active ventilation at the very end of class. So we saw that during quiet breathing, right, what we have happening, this is in my way, there we go. During quiet breathing, right, our inspiratory muscles are regulated by the phrenic nerve to the diaphragm and the external intercostal nerves. So we can see those firing. So we see our action potentials here, 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 corresponding to contraction of those muscles, right? So we see the inspiratory muscles contracting and then relaxing, basically lining up with these periods when we don't have those nerves firing, right? Our expiratory muscles, which are internal intercostals, during quiet breathing, don't have any depolarizations, right? Don't have any action potentials because they're not doing anything. We're not creating any tension using those muscles. During quiet breathing, we're just passively relaxing the diaphragm and the external intercostals, which pulls the chest back to its resting position. And the time that we see those internal intercostals and those expiratory muscles activated is during active ventilation. So during sort of forced breathing, heavier breathing, where we are trying to get more air into our lungs and therefore also force out more air, right? We can see that the lung volume here during active ventilation is higher than our lung volume during quiet breathing. And so in order to do this, we need to activate those expiratory muscles basically on the off cycle compared to the inspiratory muscles, right? So we're moving from inspiration to expiration. Okay. So that's where we left off. So we're going to continue today thinking about basically some of the reflexes that are going to be controlling ventilation and some of the receptors that are going to tell our brain how we might need to change our ventilation to appropriately match our body's demands. So when we're thinking about the neural control of breathing, right? So we have these motor neurons that we're looking at that are physically controlling the muscles, right? So we had the phrenic nerve, we had the internal and external intercostal nerves. That's what we talked about just now and at the end of Monday. Those are parts of our peripheral nervous system, right? So they're motor neurons, they're efferent, meaning they're sending out signals to the skeletal muscle. So they're a component of breathing, but they're not the whole story. We have a lot of things going on in the central nervous system. So in the brain, and particularly in the brain stem, that are controlling our breathing rate and our breathing rhythm. Now, not all of these pieces of the central nervous system and their control of breathing are fully understood. If you took a look at this section in the book, you may have noticed that your book keeps repeating a couple of times that they're kind of still an active area of research. So it's kind of exciting, actually. But we know some basics that in the central nervous system, we have neurons that are controlling basically inspiration and expiration. So they're like kind of the counterparts to these motor neurons in the peripheral nervous system. We have respiratory control centers in both the medulla and in the pons. So we can think of these kind of like integration centers. We can definitely think of something called the central pattern generator, which they think is in the medulla as well, as controlling our habitual breathing rhythm. And we're going to run through each of these in a little more detail. Okay. So peripheral nervous system, outdoing those named nerves to the specific skeletal muscles. So now we're thinking central nervous system, mostly thinking brainstem. So when we're thinking about these brainstem respiratory centers, when we say we have inspiratory neurons and we have expiratory neurons, we're saying that we have these neurons right physically within the brainstem. They're not like nerves leaving out necessarily, right? Some of them, they're physically in this region in the brain. And what we can note about them is that the inspiratory neurons depolarize during inspiration, aka when we're breathing in, we see an action potential in these neurons. Opposite is true in our expiratory neurons. So in our expiratory neurons, right, we're going to see that they depolarize during expiration, aka when we're breathing out, we see an action potential in these neurons, right? So that's why we know they are linked to each other. When we're looking at different respiratory centers, which we're going to look at in a second, we're going to see that in a lot of regions, we have a mix between these types of neurons. And there are also these other sort of weird neurons that have properties of both inspiratory and expiratory neurons. So they can't really be classified as either one. So in our different respiratory centers, we're basically going to see different combinations, different amounts of these three different types of neurons. And I'm going to tell you right now, we're really mainly going to be focusing on these first two, the inspiratory versus expiratory neurons, because those are more clear in what they're doing. Okay. So now we are looking at a picture of our brain. All right, so here we see our brain, and we're focusing in on the brain stem. Right, so brain stem, we've zoomed in. So we're going to be looking at these centers in the medulla first. So the medulla, right, is this lower swelling on the brainstem. The other place we're going to be looking in a second is the pons, which is this kind of egg-shaped lump on the brainstem just above the medulla. So that's just a reminder from way back when in anatomy lab the first time. Okay. So in the medulla, we have two respiratory control centers. They're on either side of the medulla, by which we really mean on the anterior portion and on the posterior portion. So ventral and dorsal are alternate words that correspond, at least in the brainstem, to anterior and posterior. So the ventral respiratory group, which we're going to refer to as the VRG, is on the anterior side. So this is our ventral respiratory group here. Oh, there's a red arrow, so I'll use a blue circle. So we're looking here. Okay. And our dorsal respiratory group, dorsal, if you think of like a shark, it has a dorsal fin, right? Is that fin on its back? So the dorsal respiratory group is on the back, just like a shark fin. So that's this one. So dorsal respiratory group on the dorsal side of the medulla. And they are in different sort of tracts in the brain, but we don't need to remember these for our purposes because we haven't really gone into a ton of neuroanatomy. But in case you were curious and you have been studying neuroanatomy on your own, the VRG is part of the nucleus ambiguus and the DRG is part of the nucleus tractus solitarius. But you can skip that when you're studying for your exam for next week. Okay, but we have these two respiratory groups. So when we said that we have different combinations of inspiratory neurons and expiratory neurons, we can see that when we look at the composition of the VRG versus the DRG. So in this image, we've colored our different neurons by color, right? So our inspiratory neurons here are blue. Inspiratory neurons, blue. We've colored our expiratory neurons yellow. Okay. So we can see that the VRG has this central region of inspiratory neurons in the center of the VRG. And then on either side of it, it has a region of expiratory neurons. So it has two of those. So a yellow kind of cat and a yellow tail here. The DRG is mostly inspiratory neurons, so it's colored entirely blue. It's not like fully accurate. There is some mixing of other types of neurons in the DRG, but it is mostly inspiratory neurons. So because the inspiratory neurons are hypothesized to control inspiration, right? So we think that these neurons are communicating with the motor neurons that then go out to our inspiratory muscles, right? We can think of those inspiratory muscles as ultimately, if we look at the medulla, being partially controlled by both the DRG and the VRG, right? Because we have blue in both places. Whereas our expiratory neurons, remember expiration is mostly passive, right? But when we're doing forceful expiration, when we're actively breathing hard, right, we do recruit some muscles. So those expiratory muscles have their motor neurons going out through the peripheral nervous system, but we think that they're linked up with these expiratory neurons here, specifically mainly in the VRG, right? So that's what we're looking at here in the medulla for our different respiratory groups. Okay, so next, Well, next we are going to move into the Pons, but we can look at this image first. Forgot to put this in here. So what this is looking at is kind of the function of these inspiratory neurons. So what we can see here during inspiration in these inspiratory neurons is that the frequency of the active action potentials speeds up during inspiration, right? You'll note if you look at these membrane potentials that have been measured, better pick a better color, right? They're getting closer and closer together, right? We're firing faster and faster in these inspiratory neurons. So we see this sort of ramping up of action potentials during inspiration. And then during expiration, boom, they go quiet, right? So they have this characteristic pattern where they speed up as you're breathing in. And then when you breathe out, they go silent. Okay, now onto the Pons. So before we were in the medulla, a little farther down in the body, now we're moving upwards a little closer to the brain. We're looking at the Pontine respiratory group. So the Pontine respiratory group, abbreviated PRG, is in the Pons. So we got it here. And basically it contains all three of the type of neurons that we can identify that are somehow correlated, somehow controlling breathing. So it has inspiratory neurons, it has expiratory neurons, and it has these mixed neurons that are similar to both, but a little bit different from either. Now, what we think that the PRG is doing, what we think the Pontine respiratory group is doing, is not directly controlling the breathing muscles, but we think it has some role in regulating the transition between inspiration and expiration. So that moment where you, breathe in and you're at the top of your breath, and then you switch to breathing out. We think that the Pontine respiratory group is doing something to create that switch between inspiration and expiration. We don't know exactly how it does it, but that's our idea about what this group of neurons is working together to do. Okay. Finally, that leaves us with the central pattern generator. Now, we think the central pattern generator is in the medulla. And what we think it's doing is we think it's establishing a whole respiratory cycle. So the overall patterned rhythm of breathing, right? You don't breathe at sort of random speeds. You generally have a specific pace throughout a minute, throughout an hour, throughout an activity. Exactly how it establishes this rate is unknown. They are not even totally sure. It's in the medulla. That's why it's on the slide as unknown. This is an area of active research. But we do know that there's some part of the brain that is controlling the regularity of this, right? Not just controlling the muscles, the Pons, not just controlling the switch. We know there has to be something that's setting a baseline rate. One of the current hypotheses, right, is that this works kind of like the heart potentially, that we have pacemaker cells, right? We've seen before that pacemaker cells in smooth muscle or in cardiac muscle can establish a rhythm that goes on, and we've seen that our cardiac muscle cells, right, can do that just in a petri dish. If you pulse them out, plunk them in a dish. So we think that there must be some cells that work similarly to that, somewhere in the brainstem, probably establishing the respiratory cycle. But it's basically still a big question box, which is honestly kind of cool. So if any of you guys want to go into research on breathing rhythms, here's your intro. Come back in 10 years and tell me how it works. Okay. So here's a schematic of the general idea of how we think breathing is controlled and how we think all these things are working together. I'm gonna check our time. Okay. So we're going to see that we have sensory input coming from the body, and one of our next slides is on these. But we're going to talk about central chemoreceptors and peripheral chemoreceptors. So these are basically sensing mainly concentrations of carbon dioxide, but also oxygen in the case of the peripheral chemoreceptors, also pH. So that's one of our types of sensory input that we're going to have. It's really important for regulating breathing. We also have pulmonary stretch receptors that kind of feel the inflation of the lungs. We have specific irritant receptors that sense things like smoke. And there are even links with the proprioceptors out in like the muscles and joints, mainly related to activity. So to telling your brain and your breathing centers that you're on a run and they might want to start breathing faster. Okay, so this links up with information from the Pons, information from the cortex. So the cortex is, right, the traditional brain, right, the part of the brain we think of as the brain. So our voluntary control in the cortex all send this information to the central pattern generator, which we think is probably in the medulla, send impulses to mainly our inspiratory neurons, right? Mainly we control when we breathe in. Breathing out is more passive. So our inspiratory neurons in the DRG and the VRG, then sending those impulses out to our actual muscles via those motor neurons, like the phrenic nerve, like our intercostal nerves, establishing our breathing rhythm. So this is just kind of a schematic of how this could all work together. Okay. So this is just a list of some of the types of receptors that are going to be giving those respiratory centers in the brainstem information about what's going on in the body, which is going to help us determine whether we want to breathe faster or breathe slower. So we got chemoreceptors, which we're going to talk about really in detail, pulmonary stretch receptors sensing inflation of the lungs, irritant receptors sensing smoke or dust particles, muscle and joint proprioceptors largely sensing activity levels. Okay, so chemoreceptors are super cool and super important. So your chemoreceptors are part of why, like, you can only hold your breath for so long, right? It takes a lot of willpower to try and hold your breath for, I don't know, a minute. I don't know how long exactly you all can hold your breath. I cannot hold my breath for that long. But part of the reason that's so hard is that you have these chemoreceptors detecting blood levels of oxygen and carbon dioxide. And at some point, they're going to be firing off all this information to these regulatory centers in the brain and brainstem so much that they're physically going to kind of force you to start breathing again. The impulse returning to the body telling you to breathe is really powerful. And that's fortunate for all of us. So our chemoreceptors are going to be found in two places. We're going to see them in the carotid bodies. So the carotid bodies are located basically near your carotid sinus. So we're seeing that in lab this week or last week, depending on where you are in the lab cycle. And those are your peripheral chemoreceptors, peripheral because they're outside the central nervous system. There are also chemoreceptors in your central nervous system itself, in the medulla, or more formally, right, the medulla oblongata. Okay, so we're going to see them in both places. So out in the body, near the carotid sinus, in these structures called the carotid bodies, or in the medulla. Okay, so our peripheral chemoreceptors out in the carotid bodies, near the carotid sinus, right? So here we see blue pen. So our carotid sinus, right, is the swelling, right, where we have our internal and external carotids splitting off from each other. So here we see that split. So our carotid body is going to be right basically in the split. So we can see this top yellow sort of dot. These other dots are the baroreceptors. Remember those baroreceptors from our last unit, sensing basically pressure, right? Blood pressure, essentially. These carotid bodies are now sensing chemical concentrations of CO2, mainly. Okay. So they communicate with the afferent neurons via chemical messengers. We remember that afferent means it's going back to the brain. So these afferent neurons are going up to the medulla, to those respiratory control areas, giving us this information where we can pull that information together with information from our other receptors to control breathing. So the carotid bodies are responding to largely changes in carbon dioxide. I'll circle that even bigger. Because they're sensing changes in carbon dioxide, we know that we can think of carbon dioxide as a biological acid because eventually, right, we can see that it is transformed by carbonic anhydrase into carbonic acid, which can be then broken down into our hydrogen ions and bicarbonate ions. So carbon dioxide can essentially be converted partially into hydrogen ions. And the presence of hydrogen ions, right? If we have a bunch of hydrogen ions, that means we have an acid. So the more hydrogen ions we have, the lower the pH gets. And that's really what pH is, which is why we think of carbon dioxide as an acid. Okay. So carbon dioxide, hydrogen ions, and drastic O2 changes are also sensed by the carotid body. We'll see what we mean by that in a second. But it's mostly sensing this carbon dioxide and blood pH. But in the case that you have a really precipitous drop in O2, if it gets really, really low, the carotid body will respond to that. But it has to get pretty far outside the normal range of O2 for the O2 concentration to have any effect here. Okay. So here's what we mean by that. Here's why we don't think of it as mostly, ooh, don't want to change the microphone. Just want my pen. Okay. Here's why we think of it as mostly responding to carbon dioxide. So our normal level for oxygen in the blood, in the arteries, right, is 100. Super easy, right? We want that oxygen at 100 millimeters of mercury. Okay. In order for those carotid bodies to start telling your respiratory centers that there's some kind of problem, they don't really start firing off in a serious way until we drop down to 60 millimeters of mercury as our partial pressure of oxygen. So if you were down to just 80 from your normal resting level of 100, right, this would have pretty much no effect on the carotid body. That's not low enough for it to fire off. And this is why we see the minute ventilation. The minute ventilation being, right, like how much you're breathing in per minute, how much air you're getting, doesn't really change significantly at all until you get to about 60, and then it really starts to rapidly rise. That's because this receptor doesn't really kick in until this point. We remember that the PO2 of our venous blood, right, is at about like 75, right? So essentially what we're saying here is even if your blood didn't get oxygenated at all, and you got venous blood just going directly into your arteries, that's still not low enough to get the carotid body to fire. So that would have to go on for a little bit. It would have to drop even lower than that for us to sense a problem here in the O2. So it can happen, but you have to get really dangerously low for O2 to have an effect. In contrast, very small changes to the partial pressure of carbon dioxide make a big difference. So if we're looking here, right, our normal resting level of partial pressure of CO2 is 40, right? And we can see that if that concentration, that partial pressure of CO2, even if it just goes up a tiny bit, right, we are already increasing, right? For the O2, as it decreased, we had that long kind of flat area before we increased, probably longer even than that. But right now we're increasing right off the bat as soon as we have any increase at all. So if we can see, if we got up to our venous level of like 46, right, we've already seen a fair bit of an increase in the minute ventilation. Right? So our carotid body pretty immediately is going to respond to these changes in CO2. Okay? And we're specifying here arterial oxygen and arterial CO2 because the carotid body is in the carotid, which is an artery. So what it is sensing, what it is physically in contact with, is arterial blood coming pretty directly from the heart, right? We've seen that very close relationship, the carotid, especially on the left, coming directly off the aorta. Okay. So fun facts, actually, when you reach a partial pressure of CO2 of 90 or more, right? So that's about here. This is when you go into a coma, right? So we do see a drop-off point, but this is related with other bodily functions really kicking in, really getting damaged, right? And if you manage somehow to get all the way up to 180 as your partial pressure of carbon dioxide, this directly causes death. Okay. So it's important, actually, that we adjust our minute ventilation as soon as carbon dioxide starts to rise, right? Because we do not want it to get up to coma level, let alone death level, right? So we want to pull that carbon dioxide concentration back down as fast as we can. Okay. So the carotid body, right, was one of our examples of a peripheral chemoreceptor. Now we're thinking about the central chemoreceptors. So the central chemoreceptors are on the ventral surface of the medulla. So like kind of on that same side of the medulla as the VRG. And the thing we want to remember about the central chemoreceptors is that they're actually protected by the blood-brain barrier. So they're not going to respond directly to blood-brain contents. They're going to respond to the changes we can see in the cerebrospinal fluid. So in the CSF, in that sort of fluid that's bathing and nurturing our brain and our spinal cord, we are going to see some changes in response to CO2, but a lot of things are not able to cross the blood-brain barrier. So the way the central chemoreceptors respond is going to be a little bit different. Okay. Basically, and we're going to look at an image of how this works in a second, they're not responding directly to CO2, but they're going to respond to CO2 via changes in pH. Okay. Central chemoreceptors are not at all responding to changes in O2. Okay. Really just the CO2 changes. So here we go. So here is what's happening with the central chemoreceptors. No green on this image, let's pick that. Okay, so here we're looking at the medulla, right? And here are our neurons that are our central chemoreceptors. Okay, so they can't respond directly to changes in blood pH because blood pH is essentially hydrogen ions, right? Hydrogen ions are acidity, and those ions cannot pass through our blood-brain barrier, right? So any direct changes in pH will just bounce off this wall and won't get into the CSF. The central chemoreceptors are not at all responsive to O2, which is why it's not on this diagram. CO2 can't get directly in to touch the chemoreceptors, but what can happen is that it can get into the CSF in this form of CO2 and then be converted by carbonic anhydrase into carbonic acid, and then it turns into our bicarbonate ions. This is bicarbonate, and our hydrogen ions here So what the central chemoreceptors are directly responding to is that we've turned the carbon dioxide in the CSF into a lot of hydrogen ions swimming around. And it's these hydrogen ions in the CSF that directly touch the central chemoreceptors and tell them to fire off. Okay, so what we're looking at here are some reflexes that we call the chemoreceptor reflexes that change ventilation in response to changes in oxygen and carbon dioxide in the blood and in the cerebrospinal fluid. So the easiest one to track is what's happening with oxygen. So remember, green means an initial stimulus on these diagrams. So if we have our oxygen drop down super low, way below 60, our peripheral chemoreceptors, like our carotid body, will detect this. When we have low oxygen, means we need more oxygen, means we need to breathe more. So this will create an increase in ventilation, right? So we're going to breathe more. Our CO2 is a little more complicated because we're going to be able to sense that CO2 change in both the peripheral and the central chemoreceptors, although not necessarily directly. All right, so as we have a rise in our partial pressure of carbon dioxide, one thing that's happening is that directly our peripheral chemoreceptors can sense that. So like the carotid body can see the CO2. If we have a buildup of CO2, it means we're not breathing out enough, right? We're not getting rid of this waste product enough, which means we need to breathe faster so that we can breathe out this CO2. So it creates an increase in ventilation so that we can breathe it all out. The increase in the partial pressure of carbon dioxide also creates an increase in hydrogen ions because essentially using carbonic anhydrase and carbonic acid as an intermediate step, we can turn carbon dioxide into bicarbonate and hydrogen ions. So we have an increase in hydrogen ions, lowering the pH. Peripheral chemoreceptors also sense that. So they detect that. Means the same thing, right? Still means we have a lot of carbon dioxide, still means we need to breathe out more. And a similar thing is happening in the cerebrospinal fluid. So if we have a high partial pressure of carbon dioxide, that carbon dioxide gets into our CSF. In the CSF, it is turned into hydrogen ions, aka an acid, which the central chemoreceptors can sense, and therefore tells us to breathe out. So actually what's happening when you are holding your breath, right, probably mostly, is not actually so much that your body needs you to breathe in more, right? Because you're probably not lowering your oxygen that low. What's actually happening is that your body is sensing that as you're doing metabolism, right, you're still producing ATP because your body's still functioning, your cells still have to do that. So what's happening is that your body is actually sensing this buildup of carbon dioxide, because when you're holding your breath, you're not breathing out either. So this effect where you need to start breathing again is largely due to this effect that you need to breathe out. And it can be both, right? Like after you breathe out, you do breathe in again. But that really big impulse is from this buildup of the carbon dioxide, which is why sometimes you can get away with, if you're really trying to hold your breath, you can breathe out a little bit first and then keep holding your breath for a couple more seconds, I've found. It's partially because it's this buildup of CO2. Okay. So our reflexes to respond to hypoventilation, which is not breathing enough, or hyperventilation, which is breathing too much, work pretty much the same. So In hypoventilation, it means we're not breathing enough. If we're not breathing enough, our carbon dioxide partial pressure is going up, which means our blood is getting more acidic because there are more hydrogen ions. And because we're not breathing enough, we also have this lowered partial pressure of oxygen. Our various chemoreceptors will detect and respond to this depending on the level. Right? Our carotid body can respond to all three of these if O2 is low enough. Our central chemoreceptors really responding to that CO2 indirectly when it's converted into pH in the cerebrospinal fluid. They go to the respiratory control centers, increasing ventilation. And then when you increase ventilation, it's going to reverse all of these, right? This is going to be a form of negative feedback. So now, as we breathe faster, it's going to lower our carbon dioxide, which is going to lower our hydrogen ions, which is going to raise our oxygen concentration, basically bringing us back to normal. We can also control hyperventilation in just the opposite way, basically. So if we saw the opposite thing, we had hyperventilation. So if we saw hyperventilation, we'd want to decrease ventilation as we have the lowered CO2 and we have our O2 getting really high. Okay. So our final topic is the local regulation of ventilation and perfusion. And perfusion here, we're referring to basically the blood flow to the arterioles and therefore to the capillaries around the alveoli. So this is combining what's happening in the air, in the ventilation, and in the blood, which is the perfusion. So we can talk about something called the ventilation perfusion ratio. And as that ratio shifts, your body is going to react to basically correct it so that we have the appropriate amount of air and the appropriate amount of blood in the lungs to give us our desired result. So we're going to be calculating this ratio to look at normal function and then to compare how we're going to fix that. So this is going to be under quite local control. So basically we're going to be constricting and dilating blood vessels in the lungs, or constricting and dilating our bronchioles, right? And all those tubes of air leading to our alveoli. So that's why it's local, because it's right there in the lungs. We don't have to mess with things in the carotid or things in the brainstem for this. Okay. So when we say we have a ventilation perfusion ratio, we calculate that VA, and in your book it has a dot to tell you it's a rate, divided by Q, which is also a rate. So VA, we've talked about before. VA is that alveolar ventilation, which we remember is our minute ventilation minus our dead space volume, basically, right? So to calculate it before, we saw it was VT times our respiration rate. And then we subtract our dead space volume, also times our respiration rate. Okay, so that was what VA was. That was our alveolar ventilation from before. We're not going to worry about calculating Q, because that's not our topic for now, but just know that that's basically the rate of blood flow, is the perfusion. So perfusion refers to rate of blood flow. Okay, so we want to regulate them to match each other. We want to have the rate of airflow and blood flow essentially be the same. So what we want this to equal is 1. That's what it equals under normal conditions. So here we see our alveoli when it does and does not equal 1. So at the top, we can see that when our rates are matched, when our alveolar ventilation and our perfusion are the same, this would equal 1. And that is what produces our normal concentrations of arterial O2 and CO2. So our normal partial pressure of oxygen is 100. Our normal partial pressure of carbon dioxide in the arteries is 40, right? And this ventilation perfusion ratio of 1 produces that. Now, if we have obstructions, this is going to change, right? So here we're imagining that we have an obstruction to our airway. So we can see that in these tubes leading to our alveolar sacs, right, we have something stuck, right? Maybe it's mucus, maybe it's a foreign body, but we're not getting air into this alveolus. So what we're saying there is that our alveolar ventilation then is decreasing, right? Which means we're not getting enough air, we're not getting enough fresh air. So because we have the same amount of blood coming there that we're trying to oxygenate, our partial pressure of oxygen in that blood is going to go down. And again, because we have this obstruction, we're not able to get rid of the carbon dioxide effectively. So our partial pressure of carbon dioxide is going up. The reverse happens if we have an obstruction in the blood vessel. So here we can see an obstruction in the blood vessel. So the main problem this is going to cause is that we're not going to have enough blood getting to the alveoli at all. But the blood that remains when we have this lowered Q, right? It's lowered Q, we're going to have more oxygen in that blood that is able to pass, if blood is able to pass around the obstruction at all. And we're going to have lowered carbon dioxide, right? So essentially we're having like too much exchange going on with the air in the alveoli. Okay. So ventilation is going, ventilation and the blood flow are both going to be altered if we have these changes in the ventilation and perfusion ratios. Right, so we have our ventilation, perfusion ratio, I'm just going to put that here, right? So if our ventilation to the alveoli decreases, right, this is going to give us a ratio less than 1. What we want to do is bring that back up to 1. So we have two options, and I'll kind of put them in purple, all right? Either we can just bring that ventilation back up, right? Or we can divide by a smaller number, right? So we could also fix this by bringing the blood flow rate down. Either of those could bring us back closer to 1. So if our ventilation in the alveoli is decreasing, what we're going to see in the body is bronchodilation. Right, so the bronchioles, so the actual air tubes, right, are going to dilate. So they're going to get wider. Their radius is going to increase, right? So which means their resistance is decreasing, so we can get more air faster. So that bronchodilation is this purple arrow. It's raising that alveolar ventilation. Or, and really both of these are happening at the same time to fix this, we're also going to see vasoconstriction in the arterioles. So the vasoconstriction in the arterioles, right, we're decreasing the radius, increasing resistance, therefore decreasing flow, brings Q down. Also helps us get back to 1. So the direct sort of correspondence of our gases here is that the bronchodilation, that change, the increase in the size of those bronchioles, is due to increases in CO2. The vasoconstriction is largely due to our decreases in the O2, both of which are effects of the ventilation to the alveoli decreasing. The opposite thing happens if we're looking at the perfusion decreasing, And hit undo about 1,000,000 times. Okay. So if instead we have our perfusion decrease, right? So now we're going to have a number greater than 1. We want to bring it back down. So our options, right, are that we can vasodilate, bring that right back up, or we can breathe a bit slower or breathe a bit less, right? We can bronchoconstrict, bringing that down, bringing us back to 1. So our increase in O2 under this situation, which we looked at when we looked at the blockage in the previous image, is responsible for the vasodilation. Okay. The CO2 in the bronchioles is due to the is responsible for the bronchoconstriction. So basically, you'll see, right? CO2, broncho, CO2, broncho. So what we can also notice is that it's the CO2 really that's controlling the parts of this associated with breathing. It's the oxygen that's controlling the parts related to. Okay, great. And there we are. Okay, so this is just showing us how this works. You can run through these on your own, and maybe we'll talk about it as we review, but this is not new information. This is just kind of summarizing that other stuff. Okay. So I will see you all on Friday, and we will review for the exam then. Have a great day.
Thank you.
Do you have a question for